Pre-mRNA methylation. The final modification or processing event that many pre-mRNAs undergo is specific methylation of certain bases. The most common methylation event is on the N6 position of residues, particularly when these residues occur in the sequence 5' RRACX 3', where X is rarely G up to 0.1% of pre-mRNA residues are methylated, and the methylations seem to be largely conserved in the mature mRNA, though their function is unknown. 4.5. Alternative processing. It has become clear that in many cases in eukaryotes a particular pre-mRNA species can give rise to more than one type of mRNA. This can occur when certain exons, alternative exons, are removed by splicing and so are not retained in the mature mRNA product. Additionally, if there are alternative possible poly-A sites that can be used, different 3' ends can be present in the mature mRNAs. Types of alternative RNA processing include alternative or differential splicing and alternative or differential poly-A processing. 4.6. Alternative poly-A sites. Some pre-mRNAs contain more than one set of the sequences required for cleavage and polyadenylation. The cell or organism has a choice of which one to use. It's possible that if the upstream site is used, then sequences that control mRNA stability or location are removed in the portion that is cleaved off. Thus, mature mRNAs with the same coding region, but different G stabilities or locations, could be produced from one gene. In some situations, both sites could be used in the same cell at a frequency that reflects their relative efficiencies, strengths, and the cell would contain both types of mRNA. The efficiency of a poly-A site may reflect how well it matches the consensus sequences in other situations. One cell may exclusively use one poly-A site, while a different cell uses another. The likely explanation is that in one cell the stronger site is used by default, but in the other cell a factor is present that activates the weaker site so it is used exclusively, or that prevents the stronger site from being used. In some cases, the use of alternative poly-A sites can cause different patterns of splicing to occur. 4.7. Alternative splicing. 4 common types of alternative splicing are summarized in Fig 13. In Fig 13, it is the choice of promoter that forces the pattern of splicing, as happens in the alpha amylase and myosin light chain genes. The exon transcribed from the upstream promoter has a stronger 5' splice site which outcompetes the downstream one for use of the first 3' splice site. This happens in salivary gland for the alpha amylase gene when specific transcription factors cause transcription from the upstream promoter. In the liver, the downstream promoter is used and the weaker, second 5' splice site is used by default. Alternative splicing caused by differential use of poly-A sites is shown in Fig 13. The stronger 3' splice site is only present if the downstream poly-A site is used and thus the penultimate exon will be removed. If poly-A site is used, such as in a different cell or at a different stage of development, splicing occurs by default using the weaker upstream 3' splice site. In the case of globulins, use of a downstream poly-A site includes exons encoding membrane anchoring regions, whereas when the upstream site is used these regions are not present and a secreted form of immunoglobulin is produced. In some situations, introns can be retained, as shown in Fig. If the intron contains a stop codon then a truncated protein will be produced on translation. This can give rise to an inactive protein, as in the case of the P-element transposase in Drosophila somatic cells. Germ cells, a specific factor or the lack of one present in somatic cells, causes the correct splicing of the intron and longer mRNA is made which is translated into a functional enzyme in these cells only. The final type of alternative splicing, Fig 13, illustrates that some exons can be retained or removed in different circumstances. A likely reason is the existence of a factor in one cell type that either promotes the use of a particular splice site or prevents the use of another. The rat troponin T pre-mRNA can be differentially spliced in this way. 4.8. RNA editing, an unusual form of RNA processing in which the sequence of the primary transcript is altered is called RNA editing. Several examples exist, and they seem to be more common in non-vertebrates. In it causes a single base change from C to U in the apolipoprotein B pre-mRNA, creating a stop codon in the mRNA in intestinal cells at position 6666 in the 14500 NT molecule. The RNA in the liver makes apolipoprotein 8100, a 512 kDa protein, but in the intestine editing causes the truncated apolipoprotein 848, 241 kDa to be made. Similarly, a single A to G change in the glutamate receptor pre-mRNA gives rise to an altered form of the receptor in neuronal cells. The RNA editing changes in the ciliated protozoan, Leishmania, are much more dramatic. When the cDNA for the mitochondrial cytochrome B gene was cloned, it had a coding region corresponding to the protein. Institute for Advanced Studies, Jodhpur 24. Sequence. However, although the gene was known to be encoded by the mitochondrial genome, no corresponding sequence was apparent. Eventually, some cDNA clones were obtained which had sequences corresponding to intermediates between the mature mRNA and the genome. Between the mature mRNA and the genomic sequence. It seems that the primary transcript is edited successively by introducing new residues at specific points. Many cycles of editing eventually produce the mature mRNA which can be translated. Short RNA molecules called guide RNAs seem to be involved. Their sequences are complementary to regions of the genomic DNA in the edited RNA. Several other types of RNA editing are known. B5. RNA transport and post-processing regulation. RNA export and subcellular localization. In eukaryotes, mRNA is synthesized in the nucleus and must be transported to the cytoplasm for translation. It is thought that the restriction of RNA processing complexes to discrete foci in the nucleus plays a direct role in the subsequent export of RNA through nuclear pores. The exact mechanism of export is still not fully understood. It is known that there is selective transport of mRNPs, and that export is dependent upon ATP hydrolysis. It is all known that FeedCap plays a major role in nuclear export. Uncapped transcripts such as rRNA and U6snRNA are not exported, and that the presence of spliceosome components blocks export, thus preventing the translation of partially spliced transcripts. HNRNP proteins are removed from the transcript before export, and some dissociate following transport and return to the nucleus to be reused. Processed mRNA associates immediately with ribosomes as it leaves the nucleus. RNA export from a nucleus represents a potential regulatory target in eukaryotes, but to date only viral RNA has been shown to be controlled in this manner. The best characterized system is the HIV genome, where splicing and export are controlled by the Rev protein and cis-acting elements, Rev response elements, in the introns. Rev appears to facilitate the export of RNA with bound spliceosomes, and thus allows partially spliced HIV genomes to be exported. It is not clearly understood how Rev circumvents the normal nuclear inhibition of this process. Once in the cytoplasm, mRNAs associated with ribosomes may diffuse freely, or may be targeted to. Institute for Advanced Studies, Jodhpur 25. Particular region of the cell. Polypeptide transcripts encoding secreted proteins are often transported to the membrane of the rough endoplasmic reticulum so that the polypeptide can be translocated into the lumen of this organelle signal peptide. Whereas this relies upon a localization signal in the polypeptide, in other cases the signal is carried by the RNA itself, RNA targeting. Certain transcripts become associated with the cytoskeleton and are localized to specific regions of the cell. A common phenomenon during animal development is it provides a mechanism to localize positional signals of cytoplasmic determinants in the egg, e.g. bicoid and nanos mRNA in Drosophila development, VG1 mRNA in Xenopus development. In other cases, localization can be directly related to function. Localization of alpha-actin and beta-actin mRNAs during myoblast differentiation, or may be essential for cell survival, e.g. translation of myelin basic protein in the wrong location is lethal. In each case, the localization signal is found in the 3' UTR of the transcript and associates with one or more proteins required for its localization. mRNA turnover. The abundance of a particular transcript is controlled both by its rate of synthesis and its stability, which reflects its rate of degradation. The stability of mRNA determines how quickly the steady-state levels of the mRNA change when the rate of transcription is altered, and thus how much is available for protein synthesis. Like other forms of gene regulation, RNA stability can be constitutive or regulated. The rate of mRNA degradation, which can be expressed as the mRNA half-life, can affect rapid and transient alterations to the abundance of a particular mRNA without any change in transcriptional activity. mRNA turnover and retroregulation in bacteria. mRNAs have short half-lives, in the order of several minutes for the most stable transcripts, which allows the rate of protein synthesis to be altered rapidly in response to the environment by regulating the rate of transcription. mRNA degradation in prokaryotes is mediated by RNA endonucleases, also called RNA restriction enzymes, and 3' 5' exonucleases. The secondary structure of mRNA is important in the determination of stability, with the most stable transcripts possessing multiple hairpins and stem loop structures in the 3' untranslated region which may protect the transcript from exonuclease activity. Transcripts which contain endonuclease target sites are particularly unstable. The enzymes which degrade RNA in bacteria are not well characterized, although mutations which disrupt the E. coli ribonuclease E protein induce a 2 to 3-fold increase in RNA stability. The close relation of transcription, protein synthesis and degradation in bacteria permits an unusual form of gene regulation, termed retroregulation, where RNA degradation is regulated at the level of transcriptional termination. Gene expression depends upon whether or not a cis-acting element located downstream of the gene is transcribed. If it is, the nascent RNA adopts a structure favoring rapid degradation and translation is prevented. If transcription terminates prior to this site, the RNA is relatively stable and protein synthesis proceeds. Retroregulation is used, for example, by bacteriophage lambda to control the expression of its integrase gene. mRNA turnover in eukaryotes. Eukaryotic mRNAs are generally much more stable than bacterial transcripts. The half-life of RNA ranges from 5 to -45 minutes, and metazoan mRNA is even more stable, with an average half-life of about 10 hours, reflecting the relatively constant environment of cells in multicellular organisms. The polyadenylate tail present on most mammalian mRNAs appears to confer stability by binding the PABP, which maintains tail length. Of PABP results in rapid mRNA degradation in mammalian cells, but in yeast cells, the presence of PABP appears to be a signal for degradation, so its precise role is unclear. In eukaryotes, exoribonuclease degradation of the polyadenylate tail is the first stage in mRNA degradation. Histone mRNAs, which lack polyadenylate tails, are degraded by a specific exonuclease. There is also evidence for endoribonucleases active in eukaryotic cells. Some eukaryotic regulatory proteins are required in transient bursts, and their mRNAs are consequently unstable like bacterial transcripts. These transcript many immediate early genes, the genes induced by signal transduction cascades and required to produce a short-lived regulatory responses, e.g. CFOS, CGen, oncogenes in cancer, signal transduction. Many RNAs contain specific instability elements, often a rich element spars, areas such as multiple copies of the sequence AUUA, generally located in the 3' UTR, e.g. in interleukin-1, interferon beta and CFOS mRNAs, although in several cases, within the coding region, e.g. in beta-tubulin and CMYC mRNAs. The mechanism by which instability is conferred is not understood. Rs have the ability to form stem loop structures, suggesting that factors which influence RNA folding may regulate RNA stability. Instability elements appear to be relatively independent, as they can confer instability on heterologous mRNA when inserted into the 3' untranslated region. However, there may be some dependence on secondary structure and/or context as similar elements have been identified in a number of stable transcripts such as neuron-specific enolase mRNA. The stability of a few eukaryotic mRNAs can be regulated by transacting factors which bind to the instability elements. This occurs in the transferrin. Institute for Advanced Studies, Jodhpur 26. mRNA which, in the presence of excess intracellular iron levels, is degraded, presumably by the same process which controls degradation of the constitutively unstable transcripts described above. When iron levels fall, however, a protein factor binds to an iron response element, IR, in the transcript and prevents degradation. IRs are 30BP motifs which form stem loop structures with instability elements in the stem. The presence of the IR binding protein is thought to block access to the instability elements. In other transcripts, the same IR is used to regulate translation. C1. Ribosomes, RNPs, and their study. The RNA molecules in cells usually exist complexed with proteins, specific proteins attaching to specific RNAs. These RNA protein complexes are called ribonucleoproteins, RNPs. Ribosomes are the largest and most complex RNPs and are formed by the rRNA molecules complexing with specific ribosomal proteins during processing. The method used to study RNPs, including dissociation, where the RNP is purified and separated into its RNA and protein components which are then characterized. The assembly is used to discover the order in which the components fit together and, if the components can be modified, it is possible to gain clues as to their individual functions. Electron microscopy can allow direct visualization if the RNPs are large enough, otherwise it can roughly indicate overall shape. Antibodies and individual components can be used for purification, inhibition of function and, in combination with electron microscopy, they can show the crude positions of the components in the overall structure. RNA experiments can show whether a particular protein binds to an RNA, and subsequent treatment of the RNA protein complex with RNase protection experiment can show which parts of the RNA are protected by bound protein, i.e. the site of binding. Cross-link experiments using UV light with or without chemical cross-linking agents can show which parts of the RNA and protein molecules are in close contact in the complex. Physical methods such as neutron and X-ray diffraction can ultimately give the complete 3D structure. Collectively, these methods have provided much information on the structure of the RNPs. Prokaryotic ribosomes. The importance of ribosomes to a cell is well illustrated by the fact that in E. coli ribosomes account for 25% of the dry weight, 10% of total protein and 80% of total RNA. Figure 14 shows the components present in the E. coli ribosome. The 7s ribosome of molecular mass 2.75 by 106 daltons is made up of a large subunit of 50S and a small subunit of 30S. The is composed of one copy of the 16S rRNA molecule and 21 different proteins denoted S1 to S21. The large subunit contains 123S and 15S rRNA molecule and 31 different proteins. These were named L1 to L34 after fractionation on two-dimensional gels. The L26 spot was later found to be S20. L7 is the acetylated version of L12 and L8 is a complex of L10 and L7, hence there are only 31 different large subunit proteins. Sizes of these ribosomal proteins vary widely, from L34 which is only 46 amino acids to S1 which is 557. These relatively small proteins are basic, which might be expected since they bind to RNA. It is possible to reassemble functional E. coli ribosomes from the RNA and protein components, and there is a defined pathway of assembly. The various methods of studying RNPs have led to the structures shown in Figure 15 for the E. coli ribosomal subunits. Eukaryotic ribosomes. The corresponding sizes of, and components in the 80S eukaryotic rat ribosome are shown in Fig 14. In the subunit, which contains about 45 proteins in one 5S rRNA molecule, the 5.8S rRNA and the 28S rRNA molecules together are the equivalent of the prokaryotic 235 molecule. The small 40S subunit contains the 18S rRNA and about 30 different proteins. Although the rRNAs are larger in eukaryotes, there is a considerable degree of conservation of secondary structure in each of the corresponding molecules. Due to their greater complexity, the eukaryotic ribosomal subunits have not yet been reassembled into functional complexes and their structure is less well understood. The ribosomes in a typical eukaryotic cell can collectively make about 1 million peptide bonds per second. Institute for Advanced Studies, Jodhpur 27. C2. Genetic code. The genetic code is the correspondence between the sequence of the 4 bases in nucleic acids and the sequence of the 20 amino acids in proteins. The code is a triplet code, where 3 nucleotides encode 1 amino acid, and this agrees with mathematical argument as being the minimum necessary, 42 equals 16 less than 20 less than 43 equals 64. However, there are only 20 amino acids to be specified in potentially 64 different triplets. Most amino acids are specified by more than one triplet and the genetic code is said to be degenerate, or to have redundancy. From a third point, each group of 3 bases in the coding region of the mRNA represents a codon which is recognized by a complementary triplet, or anticodon, on the end of a particular tRNA molecule. The triplets are read in non-overlapping groups and there is no punctuation between the codons to separate or delineate them. They are as adjacent triplets once the process of decoding has begun at the correct start point. As more gene and protein sequence information has been obtained, it has become clear that the genetic code is very nearly, but not quite, universal. This supports the hypothesis that all life has evolved from a single common origin. 2.1 Deciphering. In the 1960s, Nirenberg developed a cell-free protein synthesizing system from E. coli. It was a centrifuge cell lysate which was DNA treated to prevent new transcription and which would carry out limited protein synthesis if natural or synthetic mRNA was added. To determine which amino acids were being polymerized into polypeptides, it was necessary to carry out 20 reactions in parallel. Each reaction had 19 non-radioactive amino acids and one amino acid labeled with radioactivity. The enzyme polynucleotide phosphorylase was used to make synthetic mRNAs that were composed of only one nucleotide, that is poly-U, poly-C, poly-A, and poly-G. Protein synthesis took place after adding one of these homopolymeric synthetic mRNAs, then in one of the 20 reaction tubes radioactivity would be incorporated into polypeptide. In this way, it was found that poly-U caused the synthesis of polyphenylalanine, poly-C coded for polyproline and poly-A for polylysine. Poly-G did not work because it formed a complex secondary structure. If polyphosphorylase is used to polymerize a mixture of 2 nucleotides, say U and G, at unequal ratios such as 0.76, 0.24, then the triplet GGG is the rarest and UUU will be most common. Triplets with 2S and 1G will be the next most frequent. These random copolymers of synthetic mRNAs in the cell-free system in determining the frequency of incorporation of particular amino acids, it was possible to determine the composition of the codon for many amino acids. The precise sequence of the triplet codon can only be worked out if additional information is available. Towards the end of the 1960s, it was found that synthetic trinucleotides could attach to the ribosome and bind their corresponding amino acid tRNAs from a mixture. Upon filtering through a membrane, only the complex of ribosome, synthetic triplet and amino acyl trino was retained on the membrane. If the mixture of amino acyl tRNAs was made up 20 times, but each time with a different radioactive amino acid, then in this experiment specific triplets could be assigned unambiguously to specific amino acids. A total of 61 codons were shown to code for amino acids and there were 3 stop codons. Table 1. Institute for Advanced Studies, Jodhpur 28. 2.2 Features. The genetic code is degenerate or it shows redundancy. This is because 18 out of 20 amino acids have more than one codon to specify them, called synonymous codons. Only methionine and tryptophan have single codons. The synonymous codons are not positioned randomly, but are grouped in the table. Generally they differ only in their third position. In all cases, if the third position is a pyrimidine, then the codons specify the same amino acid are synonymous. In most cases, if the third position is a purine the codons are also synonymous. If the second position is a pyrimidine then generally the amino acid specified is hydrophilic. If the second position is a purine then generally the amino acid specified is polar. 2.3 Effect of mutation. It is generally considered that the genetic code evolved in such a way as to minimize the effect of mutations. The most common type of mutation is a transition, where either a purine is mutated to the other purine or a pyrimidine is changed to the other pyrimidine. Transversions are where a pyrimidine changes to a purine or vice versa. In the third position, transitions usually have no effect, but can cause changes between men and eel, or TRP and stop. Just over half of transversions in the third position have no effect and the remainder usually results in a similar type of amino acid being specified, for example Asperg-Blue. In the second position, transitions will usually result in a similar chemical type of amino acid being used, but transversions will change the type of amino acid. In the first position, mutations both transition and transversions usually specify a similar type of amino acid, and in a few cases it is the same amino acid. 2.4 Universality. For a long time after the genetic code was deciphered, it was thought to be universal, that is the same in all organisms. However, since 1980, it has been discovered that mitochondria, which have their own small genomes, utilize a genetic code that differs slightly from a standard or universal code. Indeed, it is now known that some other unicellular organisms also have a variant genetic code. Table 2 shows the variations in the genetic code. ORAFs, inspection of DNA sequences, such as those obtained by genome sequencing projects, by eye or by computer will identify continuous groups of adjacent codons that start with ATG and end with TGA, TAA or tag. These are referred to as open reading frames, or ORFs, when there is no known protein product. When a particular ORF is known to encode a certain protein, the ORF is usually referred to as a coding region. Hence, an ORF is a suspected coding region. Table 2 modifications of the genetic code unusual. Meaning. Alterative organelle or organism. Odd stop, S for some animal mitochondria AGG. A eel met mitochondria CGGR TRP plant mitochondria kunlu THR yeast mitochondria AUU lice start some prokaryotes Gugval. UUG lu. UAA stop glue some protozoans UAG. UAA stop TRP mitochondria, mycoplasma. Overlapping genes. Although it is generally true that one gene encodes one polypeptide, and the evolutionary constraints on having more than one protein encoded in a given region of sequence are great, there are now known to be several examples of overlapping coding regions, overlapping genes. Generally these occur where the genome size is small and there is a need for greater information storage density. Example, the phage Phi X174 makes 11 proteins of combined molecular mass 262 kDa from a 5386 BP genome. Without overlapping genes, this genome could encode at most 200 kDa of protein. 3 proteins are encoded within the coding regions for longer proteins. Prokaryotes, the ribosomes simply have to find a second start codon to be able to translate the overlapping gene, and they may achieve this without detaching from the template. Eukaryotes have a different way of initiating protein synthesis and tend to make use of alternative RNA processing to generate variant proteins from one gene. C3. AMINOACYLATION of tRNA, tRNA identity, aminoacyl tRNA synthetase, translational proofreading. 3.1 tRNA structure and functions. tRNA primary structure. tRNAs are the adapter molecules that deliver amino acids to the ribosome and decode the information in mRNA. Their primary structure, i.e. the linear sequence of nucleotides, is 60 to 95 nt long, but most commonly 76. They have many modified bases, sometimes accounting for 20% of the total bases in any one tRNA molecule. Indeed, over 50 different types of modified base have been observed in the several hundred tRNA molecules characterized to date, and all of them are created post-transcriptionally. 7 of the most common types are shown in Fig 16, as nucleosides. 4 of the thymidine T, which contains the base thymine not usually found in RNA, pseudouridine site dihydroridine T, and inosine I are very common in tRNA, all but the last being present in nearly all tRNA molecules in similar positions in the sequences. The letters D and T are used to name secondary structural features. Institute for Advanced Studies, Jodhpur 29. In the tRNA primary structure, there are 15 invariant nucleotides in A which are either purines R or pyrimidines Y. Using the standard numbering convention where position 1 is the 5' end and 76 is the 3' end, these are 14, 18, 19, 21, D-loop 32, 33, 37, anticodon 53, 54, 55, 56, 57, 58, 60, 61, T-loop 74, 75, 76 acceptor loop. The positions of invariant and semivariant nucleotides play a role in either the secondary or tertiary structure. tRNA secondary structure. All tRNAs have a common secondary structure, i.e. base pairing of different regions to form stems and loops, the cloverleaf structure shown in Fig 17 a. This structure has a 5' phosphate formed by RNA sp cleavage, not the usual 5' triphosphate. It has a sp formed by base pairing between the 5' and 3' ends of the tRNA. However, the invariant residues 74 to 76, i.e. the terminal 5' CCA 3', which are added during processing in eukaryotes, are not included in this base pairing region. This stern is called the amino acid acceptor stem. Working 3' anticlockwise, the next secondary structural feature is called the D-arm, which is composed of a 3 or 4 BP stem and loop called the D-loop, DHU loop, usually containing the modified base dihydrouracil. The next structural feature consists of a 5 BP stem and a 7 residue loop in which there are 3 adjacent nucleotides called the anticodon, which are complementary to the codon sequence, a triplet in the mRNA that the tRNA recognizes. The presence of inosine in the anticodon gives the tRNA the ability to be ASE paired to more than one codon sequence. Next there is a variable arm which can have between 3 and 21 residues and may form a stem of up to 7 BP. The other positions of length variation in tRNAs are in the D-loop shown as dashed lines in Fig 17a. The major feature of secondary structure is the T-arm or T-Cy-C arm, which is composed of a 5 BP stem ending in a loop containing the invariant residues GT-Cy-C. Note that the majority of the invariant residues in tRNA molecules are in the loops and do not play a major role in forming the secondary structure. Several of them help to form the tertiary structure. Tertiary structure. There are 9 hydrogen bonds, tertiary hydrogen bonds, that help form the 3D structure of tRNA molecules. They mainly involve base pairing between several invariant bases and are shown in Figure 17b. Base residues in the D and T arms fold the tRNA molecule over into an L shape, with the anticodon at one end and the amino acid acceptor site at the other. The tRNA tertiary structure is strengthened by base stacking interactions, Figure 17c. tRNA function. tRNAs are joined to amino acids to become aminoacyl tRNAs, charged tRNAs, in a reaction called aminoacylation. It's these charged tRNAs that are the adapter molecules in protein synthesis. Special involved aminoacyl tRNA synthetases carry out the joining reaction, which is extremely specific, i.e. a specific amino acid is joined to a specific tRNA. These pairs of specific amino acids and tRNAs, or tRNAs and aminoacyl tRNA synthetases, are called cognate pairs, and the nomenclature used is shown in Table 1. Institute for Advanced Studies, Jodhpur 30. Institute for Advanced Studies, Jodhpur 30. Table, nomenclature of tRNA synthetases and charged tRNAs. tRNAs or serral tRNA synthetase serral tRNAs or leucine tRNA LEU leucyl tRNA synthetase leucyl tRNA LEU tRNA LEU. UUA leucyl tRNA LEU. UUA. 3.2 amino acylation of tRNAs. The general aminoacylation reaction is shown in Fig. 18. It is a two-step reaction driven by ATP. In the first step, AMP is linked to the carboxyl group of the amino acid, giving a high-energy intermediate called an aminoacyl adenylate. The hydrolysis of the pyrophosphate released to 2 molecules of inorganic phosphate drives the reaction forward. Hydrolysis of the pyrophosphate released to 2 molecules of inorganic phosphate drives the reaction forward. In the second step, the aminoacyl adenylate reacts with the appropriate uncharged tRNA to give the aminoacyl tRNA and AMP. Some synthetases join the amino acid to the 2'-hydroxyl of a ribose and some to the 3'-hydroxyl, but once joined the two species can interconvert. Formation of an aminoacyl tRNA helps to drive protein synthesis as the aminoacyl tRNA bond is of a higher energy than the peptide bond, and thus peptide bond formation is a favorable reaction once this energy-consuming step has been performed. 3.3. Aminoacyl tRNA synthetases. Despite the fact that they all carry out the same reaction of joining an amino acid to a tRNA, the various synthetase enzymes can be quite different. They fall into one of 4 classes of subunit structure, being either alpha-2, alpha-4 or alpha-2 beta-2. Polypeptide chains range from 334 to over 1,000 amino acids in length, and these enzymes contact the tRNA on the underside in the angle of the L shape. They have a separate amino acid binding site. The synthetases have to be able to distinguish between about 40 similarly shaped, but different, tRNA molecules in cells, and they use particular parts of the tRNA molecules, called identity elements, to be able to do this. These are not always the anticodon sequence, which does differ between tRNA molecules. They can include base pairs in the acceptor stem, and if these are swapped between tRNAs, then the synthetase enzymes can be tricked into adding the amino acid to the wrong tRNA. For example, the G3, U70 identity element of tRNA-ALA is used to replace the 3,70 base pair of either tRNA-CYs or tRNA-PHE, then these modified tRNAs are recognized by alanine tRNA synthetase and charged with alanine. 3 and 4 proofreading. Some synthetase enzymes that have to distinguish between two chemically similar amino acids can carry out a proofreading step. If they accidentally carry out step 1 of the aminoacylation reaction with the wrong amino acid, then they will not carry out step 2. Instead, they will hydrolyze the amino acid adenylate. This proofreading ability is only necessary when a single recognition step is not sufficiently discriminating. Discrimination between the amino acids V and T can be achieved in one step because of the O group difference on the benzene ring, so in this case there is no need for proofreading. 3.5 Aspects of protein synthesis. Codon and anticodon interaction. The anticodon at one end of the tRNA interacts with a complementary triplet of bases on the mRNA, the codon, when both are brought together in the cleft of the ribosome. The interaction is antiparallel in nature, Fig.19. Some of tRNA molecules were found to interact with more than one codon, and this ability correlated with the presence of modified nucleosides in the 5' anticodon position, particularly inin. Inosine is formed by post. Institute for Advanced Studies, Jodhpur 31. Transcriptional processing of adenosine if it occurs at this position. This is carried out by anticodon deaminase, which converts the 6-amino group to a keto group. Wobble. The wobble hypothesis was suggested by Crick to explain the redundancy of the genetic code. He realized, by model building, that the 5' anticodon base was able to undergo more movement than the other two bases and could thus form non-standard base pairs as long as the distances between the ribose units were close to normal. His specific predictions are shown in table along with actual observations. No purine-purine or pyrimidine-pyrimidine base pairs are allowed as the ribose distances would be incorrect. No single tRNA could recognize more than 3 codons. Hence, at least 32 tRNAs would be needed to decode the 61 codons, excluding stop codons. tRNAs can recognize either 1, 2 or 3 codons, depending on their wobble base, the 5' anticodon base. If its it is only the codon ending in G. If it is G, it will recognize the 2 codons ending in U or C. If U, which is subsequently modified, it will pair with either or G. The wobble nucleoside is never A, as this is converted to inosine which then pairs with A, C or U. Table 3. Original wobble predictions. Ribosome binding site. In prokaryotic mRNAs there is a conserved sequence H13NT upstream of the first codon to be translated, the initiation code. In prokaryotic mRNAs there is a conserved sequence H13NT upstream of the first codon to be translated, the initiation codon. It was discovered by Shine and Dalgarno and is a purine-rich sequence usually containing all or part of the sequence 5' AGGAGGU3. Experiments have shown that these sequences can base pair with the 3' end of the 16 srRNA in the small subunit of the ribosome 5' ACCU 3'. It is called the ribosome binding site, or Shine-Dalgarno sequence. It is thought to position the ribosome correctly with respect to the initiation codon. Polysomes. Has begun translating an mRNA molecule, and has moved about 70 to 80 NT from the initiation codon. The second ribosome can assemble at the ribosome binding site and start to translate the mRNA. When this second ribosome has moved along, the third can begin and so on. Multiplomes on a single mRNA are called polysomes, short for polyribosomes, and there can be as many as 50 on some mRNAs, although they cannot be positioned closer than about 80 NT. Initiator tRNA. It has shown the first amino acid incorporated into a protein chain is methionine in both prokaryotes and eukaryotes, though in the former the met has been modified to N-formal methionine. In both types of organisms, the August initiation codon is recognized by a special initiator tRNA. The initiator tRNA differs from the one that pairs with August codons in the rest of the coding region. In the prokaryote E. coli, there are subtle differences between these two tRNAs, Fig. 20. The initiator tRNA allows more flexibility in base pairing wobble because it lacks the alkylated A in the anticodon loop and hence it can recognize both August and Douglas initiation codons, the latter occurring occasionally in prokaryotic mRNAs. The non-initiator tRNA is less flexible and can only pair with August codons. Both RNAs are charged with met by the same methional tRNA synthetase to give the methional tRNA, but only the initiator methional tRNA is modified by the enzyme transformylase to give N-formal methional for NAFMet. The N-formal group resembles a peptide bond and may help this initiator tRNA to enter the P site of the ribosome whereas all other tRNAs enter the A site. 5' anticodon base. Predicted 3' codon observation. A. C. G U I. U. G. C and UA. A. C. UA converted to I by anticodon deaminase. No wobble, normal base pairing. G and modified G can pair with C and U. Not found as 5' anticodon base. Wobble is predicted. Inosine I can recognize 3' A, C or U. Institute for Advanced Studies, Jodhpur 32. C4. Mechanism of protein synthesis in prokaryotes. The actual mechanism of protein synthesis can be divided into 3 stages. Initiation, the assembly of a ribosome on an mRNA molecule, elongation, repeated cycles of amino acid. Addition, termination, the release of the new protein chain. They're illustrated in figs and involve the activities of a number of factors. In prokaryotes, the factors are abbreviated as if or EF for initiation and elongation factors respectively, whereas in eukaryotes they are called ELF and EF. There are distinct differences of detail between the mechanism in prokaryotes and eukaryotes, and most of these occur in the initiation stage. For this reason, this topic will describe the mechanism in prokaryotes and the following topic will describe the differences in detail that occur in eukaryotes. Initiation. The purpose of the initiation step is to assemble a complete ribosome onto an mRNA molecule at the correct start point, the initiation codon. The component involved are the large and small rexosome subunits, the mRNA, the initiator tRNA in its charged form, 3 initiation factors and GTP. Initiation factors if1, if2 and if3 are all just over 1/10 as abundant as ribosomes, and have masses of 9, 120 and 22 kDa respectively. Only if2 binds GTP. Although the finer details have yet to be worked out, the overall sequence of events Fig.1 is as follows. If 1 and if3 bind to a free 30S subunit. If 1 and if3 bind to a free 30S subunit. This helps to prevent a large subunit binding to it without an mRNA molecule and forming an inactive ribosome. if2 complexed with GTP then binds to the small subunit. It will assist the charged initiator tRNA to bind. The 30S subunit attaches to an mRNA molecule making use of the ribosome binding site, RBS, on the mRNA. The initiator tRNA can then bind to the complex by base pairing of its anticodon with the August codon on the mRNA. At this point, if free can be released, as its roles in keeping the subunits apart and helping the mRNA to bind are complete. This complex is called the 305 initiation complex. The 50 subunit can now bind, which displaces if and if2, and the GTP is hydrolyzed in this energy-consuming step. The complex formed at the end of the initiation phase is called the 705 initiation complex. As shown in Figs 1 to 3, the assembled ribosome has 2 tRNA binding sites. These are called the A and P sites, for aminoacyl and peptidyl sites. The site is where incoming aminoacyl tRNA molecules bind, and the P site is where the growing polypeptide chain is usually found. These sites are in the cleft of the small subunit and contain adjacent codons that are being translated. One major outcome of initiation is the placement of the initiator tRNA in the P site. It is the only tRNA that does this, as all others must enter the A site. Aminoacyl tRNA delivery. EF2 is required to deliver the aminoacylternate to the A site and energy is consumed in this step by the hydrolysis of GTP. The released EF2 GTP complex is regenerated with the help of EFTs. In the EF2 EFTs exchange cycle, EFTs displaces the GDP and subsequently is displaced itself by GT. The resultant EF2 GTP complex is now able to bind another aminoacyl tRNA and deliver it to the ribosome. All aminoacyl tRNAs can form this complex with EF2, except the initiator tRNA. Peptide bond formation. After aminoacyl tRNA delivery, the A and P sites are both occupied and the 2 amino acids that are to be joined are in close proximity. The transferase activity of the 50S subunit can now form a peptide bond between these 2 amino acids without the input of any more energy, since energy in the form of ATP was used to charge the tRNA. Institute for Advanced Studies, Jodhpur 33. E. Translocation. Complex of EFG translocase and GTP binds to the ribosome and, in an energy-consuming step, the discharged tRNA is ejected from the P site. The peptidyl tRNA is moved from the A site to the P site and the mRNA moves by 1 codon relative to the ribosome. GDP and EFG are released, the latter being reusable. A new codon is now present in the vacant A site. Recent evidence suggests that in prokaryotes the discharged tRNA is first moved to an E site, exit site, and is ejected when the next aminoacyl tRNA binds. In this way the ribosome maintains contact with the mRNA via 6 base pairs which may well reduce the chances of frame shifting. The cycle of the 3-step elongation cycle has been completed, and the cycle is repeated until one of the 3 termination codons, stop codons, appears in the A site. Termination. There are no tRNA species that normally recognize stop codons. Instead, protein factors called release factors interact with these codons and cause release of the completed polypeptide chain. RFI recognizes the codons UAA and UAG, and RF2 recognizes UAA and UGA. RF3 helps either RFI or RF2 to carry out the reaction. These factors make peptidyl transferase transfer the polypeptide to water rather than to the usual aminoacyl tRNA, and thus the new protein is released. To remove the tRNA from the P site and release the mRNA, EFG together with ribosome release factor are needed for the complete dissociation of the subunits. If3 can now bind the small subunit to prevent inactive 70S ribosomes forming. Institute for Advanced Studies, Jodhpur 34. C5. Mechanism of protein synthesis in eukaryotes. Apart from mitochondria and chloroplasts of eukaryotic cells, which are thought to originate from symbiotic prokaryotes, details of the mechanism of protein synthesis differ from that of prokaryotes. Most of these differences are in the initiation phase where a greater number of EIFs are involved. The method of finding the correct start codon involves a scanning process as there is no ribosome binding sequence. Although 2 different tRNA species for methionine, one of which is the initiator tRNA, the attached methionine does not become converted to N-formyl methionine. A comparison of the factors involved in prokaryotes and eukaryotes is given in table. Prokaryotic eukaryotic function initiation factors. IF1, IF3E1F3, EIF4C, ELF6 binding to ribosome subunits. EIF4B, ELF4F binding to mRNA. IF2E1F, ELF2B initiator tRNA delivery ELF5 displacement of other factors. Elongation factors. EF2E1 alpha aminoacyl tRNA delivery to ribosome. EFTZ EF1BG recycling of EF2 or EF1A EFG EF2 translocation. Termination factors. RF1, RF2, RF3 ERF polypeptide chain release. Scanning. Since there is no Shine-Dalgarno sequence in eukaryotic mRNA, the mechanism of selecting the start codon must be different. Posits a scanning hypothesis in which the 40S subunit, already containing the initiator tRNA, attaches to the 5' end of the mRNA and scans along the mRNA until it finds an appropriate August. This is not always the first one, as it must be in the correct sequence context. 5' CCR CCA 3' where R equals purine. Initiation. Here shows the steps and factors involved in the initiation stage of protein synthesis in eukaryotes.